en Jean-Baptiste Ramond, que ve de Xile i treballa en temes d'ecologia microbiana, però sobretot en comunitats desèrtiques. Però ell, de fet, és francès, o sigui, ja us explicarà una mica per què ha anat a parar a Xile una mica, de voltes. Però dic, potser entén una mica el català, potser sí, perquè és francès i sap castellà, per tant. I ja està.
Gracias, Ana. That will be my only Spanish word today, at least for the presentation. So yeah, thanks for inviting me. It's always good to present your research. And just to explain to you a bit, and I think it's good for young scientists to know that science, life, and life in general is not a straight path. You always go in different routes. This is how it happens to me that I finished working in this environment in the Atacama today. First started doing my PhD in estuarine environments, so more like stuff that you actually work on in the Seine River, working on mercury-resistant bacteria. And then found a postdoc in Cape Town immediately after, working on those type of setups. So it's completely different. It's a wastewater treatment based on constructed wetland. Then we did a research fellowship still in South Africa at the University of Pretoria. And this is where actually began my story, my love of deserts, where we actually was managing a big project on the Namib Desert, microbial ecology. And so today, since 2019, so I'm an assistant professor in Chile, at the Universidad Católica de Chile, where actually most of my research is driven by the Atacama, as you can see. But I want to also say that actually I work on many things, including, for example, I once met an archaeologist, and now I'm actually involved in ancient DNA studies, for example. So science is part of having fun and discovering stuff and meeting good people. And in terms of this, so I started my lab in 2019 in Chile, and these are the four main themes that I work on right now. So yeah, all my slides are in Spanish, most of them. So mainly I work on desert and extreme environment and the microbial ecology and different setups. I actually have projects, particularly one with Anna, working on the effect of climate change on desert microbial communities around the world, and specifically soils. I have a very small project with ancient DNA about the coevolution of human pathogens, and now I'm starting a bit some work on the human microbiome and animal microbiome, just because it's interesting at some points. And so after an introduction, this is basically the two stories I would like to talk to you about. It's about what did I have worked on in the ecology of different niches and deserts, and also the question is how does microbial communities have evolved at short or long terms to the extreme conditions that you can see in desert. This is a classical environmental setup, but there is some microorganisms that live there. How and why? Actually, this is a good question. For the story, this is my car. That one. It was quite an amazing trip. Okay, so first, why deserts? Deserts are the most dominant biome in the world. All right, they represent, as you can see here on this map, so all that is not gray is considered the desert. Based on this aridity index, which is the ratio of precipitation over evapotranspiration, and if this ratio is below 0.65, you are a desert. So you've got different classes from dry, subhumid, semi-arid, arid, and hyper-arid, and you can see clearly that it's very abundant in terms of surface-wise in the planet. So from where I come and from where you live, it's quite clear that we both live in environments that are very, very dry. Half of the country for Chile and more than 80%, I suppose, for Spain. And therefore, it's important for us to understand a bit more how these function. And it's even more important in the context of climate change because, as you know, I think we were talking about this just earlier, it's quite warm today for January in Spain. The climate is changing, it's getting hotter, therefore there's more evapotranspiration, and you have a shift towards a more arid environment. So what happens is a dry subhumid desert is becoming semi-arid, arid, and an arid, more hyper-arid. And this is dramatic because the more you go along this trend, the less you've got vegetation cover, therefore you've got less productive environment. And this can be a problem as a global system, but also for us, just for our food, for example, production around the world, and particularly in those environments where there is a lot of people living. Oh, what's this one? Because these are the projections in terms of population, human population in the world. So you can see today, 2010 is in blue, projection in 20, 30 years is in brown. And you can see that today, already today, 3 billion people, sorry, live in deserts globally or at the margins. So that's it's a bit less than a half of the 40% of the population. But it's planned that by 2050, 1 billion more people, and particularly in those areas, will be living in those areas. So we have to understand and maybe use what microorganisms do in those environments. Oops, what happened? Because yeah, microorganisms are key for the functioning of those environments. As you can see, there's nothing, right? The reason is they're usually arid to hyper-arid. They have very, very little precipitation, obviously, very poor in terms of nutrient content. The temperatures vary both diurnally and at the seasonal level very, very drastically. Lots of UVs, for example, particularly in the Atacama, high winds. And because of this, there's a limitation of high eukaryotic life, more or less, of plants and animals. And therefore, in those environments, the main drivers of every ecosystem functioning are the microbes in general and the communities. And more or less, the question of my science in those global, very, very extreme system is how can a microorganism survive in an environment like this, where you need water for most of the reaction. So this is just to show you an example for photosynthesis. To be able to do one step, just this reaction, you need 6 molecules of water, which is quite complex to find in such environments. And obviously, it's not every organism is able to live there. I mean, this is a very important paper of the best group in the world from Alicante working on desert microbial ecology, where actually they have shown in 2015 quite easily that the more the aridity increases, you've got both the decreasing diversity of bacteria and fungi, but also in biomass. So obviously there's a selection that has occurred and leading to a decreased community in terms of biomass and diversity. So how do they do? Depending on the extreme conditions that they encounter, you will have more or less communities that are connected or not. So in the less extreme system, you will have something that is called a biological soil crust that covers the soil. It's a very nice little mat that usually is considered or described as the living skin of desert environments. But the more and more extreme you become, the more arid, the less those communities will be linked together, and you will have some colonization of specific niches. So the lithic system, so whether over the rock, like this, with epilitic, under the rock, which are hypolytic communities, and in the more extreme systems inside the rocks, which are the endolytic communities. Okay. So these are the generalities that you should, or at least now that you know, to follow my presentation. And we're going to start to talk about what I've been working on in different niches of deserts. And I like to start this always by using this picture. So I don't know if you know this. So this is Namibia. This is Sossusvlei. It's a very famous dried pan where you have those like fossilized trees, and the colors are quite amazing. But what is interesting is all those colors actually are environments that you can study. So the first one, for example, if it comes, yeah. So this little white pellicula that you see on top is a very, very saline crust that you have over this. It's a dried pan. And I'm going to talk in this talk about some studies I've been doing in salt pans, salinas, where we studied the microbial mat, but also some alytic communities. So as you can see here, maybe, so this is a cut of this rock, salt rock, and you've got this green pellicula here that actually shows the colonization of cyanobacteria actually in this environment. Then you've got this nice little orange. I'm going to talk to you about a study that we've conducted in South Africa, comparing two different systems, one hyper-arid and one semi-arid environment. And also this really low patch reminds us that there is grass, there are plants actually living in the desert, and these are very important. And we're going to talk about this specific structure just now, because this structure is very interesting. I don't know if you know about it, so it's the rhizosheets. So it's a very specific adaptation that plants from those two families have developed to survive, resist xeric stress. It's very important because in the family of the Poaceae, there's a lot of food crops that we actually eat, like maize, like wheat and everything. And these plants actually do produce this or create this type of structure when they're under xeric stress. And as you can see, the whole root system of such a plant is covered by this thing. And actually, it looks like a, well, I'm very bad with my hands, but in electric stuff, this looks like a cable, an electric cable protected by this gain, I don't know how to say that, around it. And in more details, this is the root that you can see here, and this is actually the very strong and thick pellicula of sand that is actually around it and protects the roots of the living part of the plant, the under part of it, from drying more. It's a very, very nice niche, and you can see why it's very difficult to see, but you see all the lateral root system that actually makes this very, very compact. And in more detail on the surface grain, you can see actually some bacteria growing on there. And so we did a study a couple of years ago. So the idea was, okay, what's happening there, more or less, in terms of community? So we studied 3 different plants that you have the names here and compare their communities depending on 3 different environments. So you've got the rhizosheet itself, so this very, very strong compacted stuff around the root. The rhizosphere, which is then what is influenced still by the root, but it's not actually the rhizosheet, compared to the boxen and the root system. And you can clearly see that you've got an enrichment in microorganism, at least in bacteria, from the boxen to the rhizosphere to the rhizosheet, and then a bit more selection inside the roots. And the fungi, you see the same thing, except we did not detect fungi in this particular study, but it doesn't mean it's not the case. So obviously, we had different communities, both at the fungal level and at the fungal or bacterial level in each of the different environments. With a very classical speciation or niche partitioning, which you can see here also depending on who they are. Okay, you've got different communities inside the different environments. What was interesting to see actually, so this is a bipartite network, and what it shows actually that you've got different the different plants and the roots. For example, you've got different colors. You've got different colors from the rhizosheet, so the more reddish color in the rhizosphere, the more bluish colors. And what it shows, make it simple because it looks fancy, but it's almost impossible to describe, is actually you've got a selection process that occurs from the bulk end. You've got different levels of selection, ultimately up to the root, where actually you've got a very, very strong barrier that the microorganisms need to cross to be able to enter the plant. So what you see here is actually that more or less there is a selection by the plants to select for good microorganisms, normally for them, so the plant growth promoting bacteria, right? So then we did another study, which was published last year, and the question was, okay, if a microorganisms to enter such a very protected system, I would say, in such a harsh environment, they most certainly need to compete. It's not just a nice place where everybody can come. It's a very strong competition to reach those refuge niche. So we can show that it's a refuge niche. For example, here you've got different variables that we measured in the field for temperature and relative humidity, depending on if it's on non-vegetated or rhizosphere environments, so not even the rhizosheet, at different periods. So what you can see is in terms of humidity, inside the plant, whether it's at the surface or at the deep, you've got more humidity. And the opposite is the temperature is lower under the plant. Okay, so it's clear that this environment is a much better environment than the outside soil. So that's the study. So what we first showed using metabolic coding, same story as last time. You've got a selection of different communities depending on the niche that you study. So from the non-vegetated to the root tissue. So nothing very fancy here. Then using Cloudprint networks, what we were able to show, if you compare just the soil, because the root system is a bit different, is that actually you've got, the closer you go to the plant, so from the non-vegetated to the rhizosheet, you've got a much more stronger connectivity and centrality, and the heterogeneity decreases. What it means is this environment have more structured community, more relaxed community. It's better environments than this one, which is more complex. The network is more loose. And you can actually see this also by the number of nodes, so the number of organisms that actually are within those networks. You can see that in the roots there's almost nothing, but you've got some kind of increased number of microorganisms within, for example, the rhizosheet, compared to the other environments. Take a look here. Not that one. But that doesn't answer our question. Is there a fight to answer this refuge niche? So to study this, we use shotgun metagenomics, and we compare the different. So here, plant growth promoting capacities, so biofertilization, promotion, protection, and competition, biotic competition, and we compare the different environment of the plants, so non-vegetated soil, rhizosphere and rhizosheet. And clearly, just looking at this, you can see that you've got kind of two different environments, so the plant impacted and the non-plant impacted. And for example, you've got much less of this section, so the bioprotection section within the soil than within the plant. You get much more of these genes of biofertilization close to the plant than the opposite. Here it's a bit different. So, and here it's very clear that you've got much more biotic competition genes within the rhizosphere and the rhizosheet than without, so all those Cas genes. And what was also more interesting to show is that actually, if you look at so this black little box that you see here, which are the genes encoding for the production of antibiotics, you got much more within the rhizosheet than within the rhizosphere, and almost nothing in the non-vegetation soil. So that means that really, to enter those environments, the microorganisms are competing against one another to then provide the plant with what the plant wants. So in very short, this is a little model that we made that actually resumes more or less what we've seen. Is actually what you have next to the rhizosheet is more linked, or there's a high biotic stress when compared to the outside, where you've got an abiotic stress that actually is the most dominant stress for environment. So you get all this oligotrophy, blah, blah, here's competition, everything. If you want, the paper is out, I can give it to you if you want this in more detail. So that's the first story. Second story, so we're going to go to South Africa this time. So the previous one was Namibia, this time it's South Africa. So we compared communities from two deserts that are the Namakwa and the Rietstelsveld. And we wanted to actually study if this had an impact on the nitrogen cycle, more or less. And our hypothesis was that in the hyperariid soils, you had much less phylogenetic and functional diversity than in the arid soil. It's based on a classical hypothesis. And this I'd like to highlight. This was a story led more or less by Thomas, which is a pregrado student of mine. The University in Chile, and with collaborators from Beatrice's group, so Jaime and Javier. And so yeah, so this is just a map that shows you that more or less you've got less nutrients in desert plant environment, and particularly nitrogen. Okay, this is just an introduction. Oops. And effectively, it's what we saw also. So we had much less nitrate in the hyperarid than the arid, and the same for the ammonium. So actually, the environmental, the study setup was good. And what we did first was we studied the diversity. So this is 16S prokaryotic. This is fungi and hyperarid. Oh, so hyperarid. This is hyperarid in the color, arid in the yellow, and you've got the fungi and 16S. So the prokaryotes, and you have the same thing. Those communities are quite different, a bit less for the fungi, but more or less you've got a nice separation of the communities depending on the environmental setup. We also did some network analysis, so these are very fresh results, but same kind of story than before. You've got here in the semi-arid environment, and here you've got the hyperarid environment. It's very clear that one is a much more strong network, has much more co-occurrences, and therefore the community is much in a better state, I would say, than this one, which is much more stressful. So we were sure more or less that we did a good sampling. But then looking at the nitrogen cycle, why did you go there? It's very simple. So it's a cycle, but you can separate it in two. So there's a part of nitrogen input, there's a part of nitrogen loss. And if you look, only the black arrows are the abiotic processes, all the other colors are biotic. And where you see genes, it's where the microorganisms actually are involved, microbial communities. So if you look at the nitrogen cycle, almost every step of the both input and losses parts are actually have microorganisms involved, and in some cases only microorganisms are involved, like nitrification, for example. And so what we did is, okay, we're going to study the nitrogen fixing capacities. So we went for, so these are the different steps that we want to study in the shotgun metagenome. And what we did was first to actually quantify the different genes. So here you probably can't see it, but you've got the one for nitrogen fixation, so NIFD, NIFH, NIFK, nitrification, etc., etc. And we compared their abundances depending on if they're arid or hyperarid states. So more or less, not much differences. I mean, so the aridity doesn't seem to affect really the potential fixation of the microbial communities within a soil of a dryland. What is interesting, actually, if you then assign those reads, or those reads, to whom they belong, you can actually see that depending on where you come from, you don't have the same, depending on the aridity, you don't have the same community doing the same function. So just as an example, if you take this gene, you can actually see, for example, that this orange part is present in the hyperarid and not in the arid. Okay, so that's the idea. So what it shows is that you have the aridity has an impact on who can perform the function. And more interesting is actually that you had a high redundancy. So that means that more than one microbial type is able to participate in one of those steps, which is actually a good news for the environment in general, meaning that if one member of the community able to do this process disappears, you've got other guys that potentially can participate or take their places, basically. And so these are basically the general function. Very general conclusions. Very, very simple. You've got a nice niche partitioning in terms of diversity, but you've got a high redundancy, functional redundancy. And this is like a result that actually Thomas sent me probably Friday. And this shows you the same stuff at other levels. So I just talked about the nitrogen cycle, which is here on this slide. So we went a bit further. So we have the same thing with the sulfur cycle and the carbon cycle. And you can see that there's a lot of different members. Like, it's just at the phyla level. So already we have a lot of different members of different phyla able to perform a lot of those different steps. So it goes, yeah, that's the story continuing right now. Hopefully papers. Right. So now I'm going to expand a bit more the study. So here, this project was born during the strong quarantena that we had in Chile that lasted a year and a half. More or less. So we couldn't go to the lab, we couldn't go to the university or whatnot. So Natalie, my PhD student, which was starting, we had to find her something to do. And so it was very simple. We downloaded shotgun metagenomes from different places around the world. So from cold and hot deserts, because yeah, I forgot to tell you about this. And so yeah, let's see what we can do with that. Because I mean, you have to sort of these are the numbers of different shotguns that we had from different environments. And what we did, part of the study was to recover metagenome assembled genomes. I don't know if you know about this. So if you sequence everything, basically you're able to reconstruct genomes one by one. And it's a very powerful tool because it allows you also to reconstruct genomes from the clades that have no cultural member, for example. And if you look at, these are the repartition of the different mags that we were able to, that she was able actually to generate. And you can see that you got a lot from Antarctica, but that makes sense because we had 35 shotgun and then we got a couple of them. And once you've done this, you have to see what is the interest of those genomes. And first, the first thing that you have to do is actually, okay, see what is novel, not the first thing, but what are the things that you have. I think I forgot to say something. Yeah, that was the thing. You have to check for the completeness and contamination. All right. So in terms of, I think I went completely the wrong way. Never mind. Okay. If you take, well, So if you take what we have, those 750 new, well, not new, metagenome assembled genomes, but they actually, if you can see, there are like lots of bacterial phyla and 2 archaeal phyla. And what was striking, I think, was actually that we were able to potentially describe 12 new families, but only in the bacterial domain, up to 152 new genus and almost 600 species, new species. So it's quite a powerful tool to actually go and investigate the diversity, both at the taxonomical level, but also at the functional level. Because now that you have the genome, you can actually study what they're able to do. And so then this is a tree of those genome of the mags from the bacteria that actually are over 90% complete and have very little contaminations. That means that we really have almost the complete genome theoretically. And as you can see, there are the new species and the new family or whatnot. They are They are in very different areas. So I've highlighted here in green. So the one from the Atacama, because obviously it's important for us. You can see that, yeah, we scatter a lot of the tree of life, at least at the bacterial level, and that's very interesting. And so we're actually right now studying the functional capacity of those guys. And we did the same thing for the archaea, so we've got much less archaeal mags. I think it's like 13 or something like this. But still, they belong to two phyla, so Bacteriota and Thermoproteodas. And also they come from different deserts, or cold and hot environment, and quite a good signal. So we're investigating those guys right now. So now a bit of salt. So this study was done. Go back to Namibia, and the idea was to study 2 different salt pans. So you have pictures here that are in the gravel plain of Namibia. So they're 120 kilometers apart, and the idea was to study what type of selection do you have in the same environment, very distant. All right, we studied, so as I said a bit earlier, so the microbial mats that you had at the surface of the map here, but also 2 different type of highlights that we recovered. So one is green and the other one is red. I don't know if you can see it very well, just for interest. And we studied them using shotgun metagenomics again to actually see what was their functional capacity, how they differ, and also their structure. This is what I'm presenting here. So we took all the 16S, and then we checked what they were in there. And so you got 2 sections. So here you have the highlights, and here you've got the microbial mats. First thing first, one is dominated by proteobacteria, and the other one is dominated by archaea from the Archaeota. All right, so that's the first thing that we have to notify, to remark. And what was very interesting is up to 20% of our community we couldn't actually have a match to. So there's still a lot of novelty that we could investigate there. And then we looked at the different cycles. So here it's a very simplified carbon cycle where we compare the microbial mat to the highlights. If you see a black arrow, we detected the function, the section, the cycle part, and if you have a gray arrow, we didn't detect it. So as you can see, you've got different sections that are, for example, present in one, absent in the other. All right, so that's the first thing. So not every community functions the same way, and particularly as I noticed, the anaerobic carbon fixation is exclusive to the microbial mats. If you look at the carbon cycle here, so you've got different abundances, obviously. What is interesting is here it's the cyanobacteria and the alpha and beta proteobacteria that are involved, while here is the cyanos and the gammas. Okay, so you got all the same type of fine differences. And then on the one side, so in the microbial mat, the proteobacteria are the ones that actually are participating the most. While if you look at the highlights, is the archaeas. So you've got a nice match between the phylogeny I showed you before and the functional level that we were investigating right now. Here it's the nitrogen cycle. So you can see that in the microbial mat compared to the highlight, it's very clear that one is almost complete and one is almost, there's almost nothing happening there. All right. Particularly, you got no nitrification here, you got no DNRA, absolutely no denitrification, etc., etc. While here, no fixation. While here, you got most of it. So here, what it shows is actually only this one is able to fix nitrogen from the atmosphere, but here, what it shows is actually nitrate is probably the most dominant form of entry, but here also, of nitrate, of the nitrogen within those communities. And while here you have still proteobacteria involved in both of them, here you see that the planktomycetes are actually involved in most of the cases, and as before, halides are also very, the planktomycetes actually are replaced, more not replaced, but actually the archaeas actually dominates. So in terms of conclusion, this first part of the presentation, so we can say that actually we have microorganisms in all of the niche and more microenvironment we studied, and they're adapted to live there. That every communities in each of those environments is actually quite rich in terms of diversity and capacity. But obviously, depending on what you study, you have different communities. And what I think is the most striking result here is actually there's a lot of strong competition to actually colonize those favorable niche in those very extreme environments. So now I'm going to talk about step 2. So how desert communities, so we're going to talk about soil only this time, adapt to those conditions at short and long term. So this time we're going to go only exclusively in the Namib Desert, because well, that's where I studied most of did most of my research, and it's interesting in this sense is it has been a desert since at least 43 million years old, since 3 million years. Okay, and most of its section is hypoarid since at least 5 million years. So obviously the microorganisms that have colonized this environment have adapted to those conditions for a long time. That's why you have this possibility to study the adaptation of microorganisms to dry conditions over long periods. So we're going to be in this environment next to this very nice research station, in this environment, which are the gravel plains. And I'm going to talk to you about 2 studies. So one is 2 metatranscriptomic studies, where we first studied what happened over a couple of days in dry soils at different times of the day, to see if actually you had microorganisms doing stuff. Was it at night, at morning, or we don't know. And then another experiment where actually we wetted the soil and measured the adaptation or what happened to those microbial communities over a very short period of time. There we go. So first thing first, so that's the first study. So first we're going to look at this little part. So this is showing you the percentage, which you don't see, of the reads that were actually detected in the metatranscriptome of the different clades. So most of them are bacteria. You've got a bit of fungi here with this red little color, and we could detect also some archaea with those little blue flags. So actinos and proteos were the dominant, which is normal because it's always the one that dominates the communities. But still, what's interesting to see is you had quite a diverse community alive during a dry period in the desert, where the life is actually active. And if you then look at different sections of the different cycles, you can actually see that you have also quite a few. So the red colors are you've got more counts, blue you've got 0. Some of those sections you have quite a few actually redundancies still at the active level with the RNA. You've got the same thing here. Interesting, but what we could show is actually compared to the metagenomic studies, if you take some specific sections of those cycles, some are actually exclusively expressed, at least active, or those that are active actually belong to 1 or sometimes just 2. Well, here it's the same family, but to its order. Here from one of the other family only. So if, for example, this could be problematic if others microorganisms are not able to be active during the day in those environments, for example, if they disappear. What we were able to actually confirm using metatranscriptomics is actually more of the nitrogen is not fixed via the atmosphere, because we almost detected no genes, but rather through nitrate and everything, which makes sense because it's readily available in those soils thanks to actually the evaporation. If you look at the carbon cycle, so this is the photosynthesis, what you can see is you get very, very little expression of those genes when compared to the other ones. So that means that more of the carbon is fixed via a chemio-autotrophic pathway or reassimilation of carbon dioxide. Then we constructed this functional network. And so in short, so you've got 4 modules, which is part of it is interesting. But what is very interesting is that all those modules are connected by a very different specific gene, and those genes are actually, turns out, are part of very, very important pathways or assimilations of nutrients. So based on this figure and what I said before, what you can say actually, even though the microbial communities in those environments are actually resisting most of the time to the stress, they actually structure their activity around the nutrient acquisition, which is actually shows actually it's a living environment, it's a living community. It's not just a resisting community so it definitely has adapted to long-term dryness. And so then we wanted to evaluate, so what happens if you stress them? So water is obviously a stress in deserts, and it's not contaminating, so it's good. So we studied over different, so just over 7 days, what happens if you water a soil. So this is one of the environmentals, this is one of the setup, we had a control next to it, and you can see we watered at different spots, and then we measured different things and at different times. So T0, 10 minutes, 1 hour, 3 hours, 7 hour, 1 day, and 7 day, and also that was metatranscriptomics. And these are the results. So this is the water content in the soil, so dry soil, no water, measured by so eye buttons. And here you can see that after the watering, you've got, so you almost reach, so you've got a high water content. And after day 3, almost no water anymore. So it dries completely very, very fast. Okay. Then what happens, you can actually see that all the dry soil communities, if you look at more or less at if you look at the community or the community itself, they're all in the same area. The dry, so those like grayish and brownish dots are in the same area. But if you look at the watered system, you can actually see that you have a shift, and then it goes back to the initial setup. All right. So mimics kind of what you see. So really water activates something, and some microorganisms and some activities is happening. And so what we detected is with water, you had different types of genes that were actually significantly activated, particularly those of motility, transport, and growth. So microorganisms eat, move, and try to colonize new niches, while the ones that are actually always present, that actually are the most so downregulated by water, or that upregulated in dry, are the one for resisting the dryness of the environment. If you compare those two setups. And so then we wanted to say who is activated actually. So same thing in the control, you've got this type of nothingness, more or less, things are happening. But then if you put water, what you have is you've got some peaks of different classes at different stages. So if, for example, you've got a very strong alpha proteobacteria peak immediately, so this is 10 minutes after the watering, and then it decreases drastically. If you look, for example, the delta, they grow until they disappear. And you've got different things like this. And for example, the eukaryotes, they basically have a little peak 3 or 7 hours after the water. So the activation of the community depends on which taxon you're talking about. And the shifts are rapid no matter what, because I mean, most of them occur within the first 7 hours. But after 7 days, you reach back to the equilibrium of the first dry condition. So with this, we basically designed a very interesting model, I think. So if here in the condition of hyperaridity, what you have, so these are the sand particles or soil particles, and what you have microorganism spores. They have very, very little. They don't do much basically. They fix a bit carbon. But then when it comes water, then you've got a connectivity process that happens, that all the grains are connected, and then so microorganisms are able to then colonize and go to new places. So I didn't show you, but we have results on the viruses also that change in terms of abundances depending on the water. So you've got more competition, you've got more everything basically. And then little by little, so once the microbial communities have grown, you've got the predators that actually come into play. So you've got the amoebas, you've got different things like this, or so some eukaryotes that are able to actually come and eat the newly developed communities. And little by little, so one day after, because this basically happens in the first 7 hours, you begin to reach a new desiccation step, and that's the cycle. So in terms of conclusion on that part, what we have, so you have two different setups. So the long-term and short-term adaptation. So first things first is in both levels, you've got the communities that are very active and very diverse. No matter what, communities are able to react and they have adapted to the environment. You got a high level of transcription of genes linked to the chemiotrophic and carbon fixation, but almost nothing linked to photosynthesis. So remember, as I said at the beginning, 6 molecules of water is probably too complex to get in such an environment. So you only do this when you can. Same thing, nitrate is probably the most feasible way for nitrogen to enter those environments, just because to break the nitrogen, it's 3 strong bonds. So obviously this is a lot of energy for microorganisms at the very level in such environment, which is very poor in material. And yeah, more or less, then you've got different mechanisms. And what was interesting to show is you've got the very active model of dispersion, the predation that happens once you've got water, which actually is an impulsor of life, no matter what, in those environments. And I just wanted to finish more or less on this slide. So this is not from my research, but it's from lots of people I know. And if I ask you this question, for you, what do you think? What is the most available food and energy source on the planet, no matter where you are? I have an answer to that. The 1 hour is difficult. It's everywhere. So obviously, microorganisms want to use it because it's easy. The idea? Gas, air, oxygen, whatever. But you've got those type of gas, trace gases. So carbon monoxide, the hydrogen, and methane. For example, so these represent, so they're trace gases in the atmosphere. These are more or less their concentration, very, very low. First thing first, 2 of those are very, very important greenhouse gases. All right, but if you look here, so it's probably difficult to see for you, but there are some enzymes that enables microorganisms to oxidize those, right? And yeah, let's say this. And so there are hydrogenases, for example, and what is interesting, for example, if you oxidize the hydrogen, theoretically, you can produce water. So this is like a very cool stuff that's a group from Chris Greening and Don have been working on. And well, he's been more for quite a long time. Actually, it shows you that actually the plasticity and things that we know on microbial communities is still something that we're trying to understand. There's new stuff almost every year, I would say, that actually changes your way of thinking, because this is quite fantastic in a dry environment. So this is Antarctica, so it's dry, it's not but I know they found those enzymes in hot environments now. Right, well, this is just to show you actually so my 2 stars of the lab, so my 2 PhD students, so Barbara and Natalie. So one is working on this rhizo sheet, but in Andean plant, because that's never been studied, and we can actually find this plant from the Atacama up to Santiago, so from a hyper-arid to a semi-arid environment. So it's a very nice environmental setup to actually study the adaptation of those plants and these communities attached to this structure in all those environments. And then, so Natalie, she's working actually on more climate change in deserts, soil desert environment. And this is more or less her sample site. So it goes from Iquique, the north of Chile, up to Antofagasta. So it's quite interesting stuff. So this is done in collaboration with the King Abdullah University of Science and Technology in Saudi Arabia, and this with with Arin and with Don and Beatrice of Chile, South Africa, Swiss. So it's really, yeah, very nice science. And about this is my group right now, small, but with more people. And I would like to thank all those people that were involved directly or indirectly in the research I've shown you, and all the funding members. And thank you for your attention. That's it.